Thank you for giving the opportunity to present my study. I am from Amala Medical Institute, which is a teaching hospital of 1,200 bed hospital, and I am professor of microbiology. So to introduce, I am going to present on NTS, non-typhoidal salmonella. Non-typhoidal salmonella belongs to Salmonella enterica, subspecies enterica, and which is considered as an agent of food poisoning of animal and poultry origin. Infection is usually self-limiting, but it can produce serious complications like septicemia in some patients, particularly in immunologically compromised patients. They are gram-negative motile bacilli belonging to Enterobacteriaceae. There are two species like Enterica, Salmonella enterica and Bongori, but Enterica can cause infection in humans as well as animals. There are typhoidal serotypes and non-typhoidal serotypes. And in non-typhoid serotypes, there are more than 2,500 serotypes, like Salmonella typhimurium or Salmonella enteritis, like that. And the aim of the study to identify the rise in incidence of non-typhoid salmonella infections following COVID-19 outbreak. We noticed in our center the samples which could isolate a huge number of non-typhoid salmonella after this COVID-19 outbreak and relaxation of the there was no to identify the next one, the probable causes for the increase in incidence of infection. So the study was designed as retrospective study of the stool samples from acute diarrheal cases received for culture by the microbiology laboratory in our center. The period January 2015 to December 2022, following this COVID-19 outbreak. The inclusion and exclusion criteria, all samples which were positive for NTS were included. The samples with the growth of other enteric pathogens and normal enteric flora were excluded from the study. Study procedure, stool samples from patients with acute gastroenteritis were collected as per standard procedure. The demography as well as comorbid conditions in the patients were noted. The NTS were isolated and identified according to standard procedure. The antibiotic resistance of the pathogens also studied. Statistically analyzed by using percentage. We calculated the number and presented in percent. The studies like that. The prevalence of NTS infection from 2015 to 2022 and compared the prevalence. We divided into three epochs, like 2002 to 2015 to 2019 pre-COVID period, 2020 like COVID period, and after the COVID like 2021 and 2022. So this is the, you can see in 2015, we got only 24 cases of NTS isolation. It comes about 5.76%. And all following years, this 16, 17, 18, little rise, 25 in 2019. And in 2020, what happened? Our hospital was changed as COVID-19 treating hospital, and number of patients coming to our hospital with other ailments decreased. So only a few number we received in our laboratory, it comes about 273, and the isolation rate is only 15%. No, 5.49%. It is just like other previous years, 18, 17, like that. But in 2021, even a low number, with a low number of samples, we could isolate 33 NTS from 286 samples. It comes about, the rate is about 11.54%. The trend is same in 2022. The number of the samples increased as well as number of isolates increased. The percentage is 11.36 again. Age-wise distribution, the demography to present, the people, majority of the patients belong to 51 to 60 years. Comorbid conditions, mainly malignancy. Ours is a malignancy treating hospital also. Chronic liver disease, chronic kidney disease, Crohn's disease, and no, without any comorbid conditions, it comes about 156 isolates. And to talk about antibiotic resistance, we could not find much resistance, rise in resistance among the isolates. For ceftriaxone, it was only 5% before COVID, pre-COVID period, 2015 to 2019. And cotrimox, that these are the primary antibiotics we are giving for this one. Ciprofloxacin only showed 27. Now, among 107 isolates in 2021 and 2022, again, in the case of ceftriaxone, the number of resistance reduced, but other also almost the same, that of pre-COVID period. To discuss, the isolation rate of NTS is low from 2015 to 2019 in our study population. Our state is Kerala. Not much data available on NTS infection. There is a study from southern part of our state. They studied 2 years, 2012 to 2014. Only 15 isolates they got. In our tertiary center, this isolation rate in that study is also same as 4 to 6%. In our study, 2015 to 19, it is only 4 to 6%, and 20 also 5.5%. But a double increase is seen in 2021 and 2022. So what are the reasons for this increase of NTS infection? Physically, we can say that this relaxation of the lockdown, 2020, there were the lockdown implemented, and 2021 Omicron wave occurred. But after that, so many celebrations and feasts occurred in our state, and people were exposed to more infections. That may be one of the reasons, but we have to evaluate the role of immune dysfunction by virus infection, because majority of the people in our state were infected by this virus. So to talk about the source of infection, why we get infection, by using the contaminated food products, because it is a zoonotic infection. We get infection from mainly poultry, meat, and eggs, as well as infected animals. And contaminated water can also spread the infection, but in our place, it is the possibility is very less, and person to person can also spread. And other sources are like unpasteurized milk. That is also usually we are using pasteurized or boiled milk for consumption. And vegetables that have come into contact with the manure may be contaminated with this bacteria, which can also spread the infection. But main source in our assumption is the contaminated food and food products from poultry, meat, and eggs. particularly mayonnaise. So there is a study from Kerala on occurrence of Salmonella enterica species throughout the poultry processing lines. They got a 67.19% the contamination rate. So we can say that it is a major source of contamination. And needless to say that more celebrations and feasts during immediate post-COVID-19 period also led for this increased infection. Then what about the possible immunological dysfunction? We know that NTS infection leads to T cell activation and antigen-specific CD4 T helper cells are activated in the gut's lymphoid tissue. The bacteria has to evade all the microbiota, the resistance mechanism by the microbiota, as well as the immunological resistance by gut's lymphoid tissue. The CD4 T cells can produce interferon gamma, which is vital for activating the macrophages. These bacteria are facultative intracellular. They can enter the macrophages, but macrophages will fail to kill the bacteria. So the number of total T cells, there are studies, CD4 and CD8 are dramatically reduced in COVID-19 patients. Can be so in the gut lymphoid tissue, the might have reduced these CD4 cells, and the survival of NTS in salmonella containing vacuoles increased. They adapted to host derived metabolic products, and they could invade. And we also analyzed this the trend in 2023 and 2024. More number of invasive infections occurred. More number of invasive infection means the septicemia, the blood isolate. Also number increased in 23-24. And usually we get these isolates from patients with comorbid conditions like malignancy, CLD, and CKD, like that. So these patients and also immunocompetent patients, there can be, we assume that there can be a temporary reduction in the CD4 counts as well as some functional reduction also might have occurred. That may be one of the important reasons for or causes for the rise in NTS infection. So conclusion, there is a transient reduction of CD4 counts. More studies, it needs more studies. And also that in future also we can anticipate a more viral infections outbreak, and these viruses also can lead to CD4 reduction in CD4 counts, and more infection can occur with the NTS. In that case, we have to look for the vaccines. Otherwise, there will be, because after this pandemic, what we see that the comorbid conditions like diabetes and respiratory infections and all are in increasing trend. So, but it needs a more study anyway. So to reduce the NTS infection, we have to look for vaccines. Okay, that's about. So these are the references. So we could publish this one. Thank you. Thank you for the patient listening. Okay.